Alright, hello, welcome back to 2. If you're seeing this and it's late, probably go to bed, but for me, I'm built different, so I'm gonna stay up for a tiny bit more before bed. I'll get cap locks on. Okay, how's everyone doing? Right, let's talk. How's how this is gonna work? All I'm gonna do is basically just do this required practical osmosis. As said on the stream. We're not going to take too long. How was the lit paper today? This might be my final stream because I just think it's helpful because I get to talk it, and then by talking, I can also teach myself it, make it more memorable. So yeah, the lit paper, I'm not going to say too much about it. It was quite good for me, but the grey boundaries are going to be astronomically high. Bit concerned for me there, but anyway, let's get on to osmosis. So I wrote a couple of notes down about it, so it'll be useful. So about osmosis, it determines like the sugar concentration in potato cells, and this time it is a. So you use the equipment shown in Figure 1 to model the effect of amylase on starch. Oh dear. Um. Y'all, I might need to retitle the stream. I've actually got the wrong questions. Okay. Let's see. I'll do this on the fly and talk about the lit paper a bit more. So the lit paper, I think it was nice. Guilt is such a open theme. It didn't bother me. It just annoys me that the grey boundaries are just going to be so high. But yeah, I had a plan to do the supernatural, the predicted stuff, but that's fine because then all I did there was pretty much because guilt was so open, I could just kind of link it, but then say, "Oh, but the punishment they get is guilt." So it wasn't too much an issue for me. Was this enzymes, right? Enzymes. Enzymes optimum temperature. And there's a rubbish thumbnail as well, so I'm gonna just change that. Bam. Okay. Yeah, guilt was nice. Christmas, everyone say, "Oh, that's such a horrible one." Literally, all we had to write about was like Dickens wants Christmas, the idea of Christmas to be carried all year round, basically, something like that. Ain't that bad? Ain't that awful? Oh my goodness, no. Right. There you go. Okay. We're getting somewhere. Save. Okay. Nice. Right. So how do I do it? I have to do this upside down, so bear with me, everyone. Or do it sideways, that way we both have to crane our necks. I'm going to do it this way because I can kind of see it, you can kind of see it. So enzymes, the aim is to determine the optimum pH of an enzyme. You draw these whole little graphs where you have like all these points. And then you kind of draw this, and this point gives you the optimum temperature. Do I think it's going to come up? I have no idea. Hopefully, otherwise this whole stream is pointless. But yeah, that's the graph. I'm not sure if you can really see it. Last time I noticed the last stream you couldn't really see it, so oh, gee. There. You kind of get it. Yeah, that. So that's what one of those graphs would look like. Here, a student has used the equipment shown in Figure 1 to model the effect of amylase. Amylase uses iodine. Iodine, you can do starch tests. But yeah, it's an enzyme, which pretty much gives you all you need to know. It's just an enzyme of starts. Bonus points, blue back. There's blue back of starts. Next I have the food tests here. And it'll be orange if not. Okay, so there we have the beaker, the water, whisking tubing. I'm guessing that's something random. It could be potatoes, it could be carrots, it could be broccoli, I don't know. At 1 minute intervals, the student uses a pipette to 1 minute intervals, that's time. Use the pipette to remove a small volume of water from the beaker. It then makes a Benedict solution in a test tube, which is then placed in a hot water at 60 degrees Celsius. The first 4 samples, there's no change in the colour of Benedict solution, but the fifth sample, the colours Benedict solution changed from blue to red. Explaining what this colour change indicates, so this one is pretty straightforward. Blue to red is the test for Benedict's solution, and that shows that there is sugar available. So if we know that pipette just water. So from what I see, the amylase is breaking down the starch and turning it sugary. So therefore, what does this change indicate? It indicates that the amylase, I don't know how to spell amylase. Amylase has broken down the starch into sugar because the Benedict. I think of Benedict Cumberbatch when it's a Benedict solution. I feel like Doctor Strange holding some weird vial. That's what I think of. Solution turns red, indicating sugar's presence. Yahoo! The food test this as well. Y'all see that? Also, read my handwriting. Hopefully, you can. Student want to test the water beaker for starts. Explaining the test and following how the student knows if the starts are present or not. The water. So this is just test for starts. Again, same kind of thing. For this one, it's iodine. Like I said here, iodine, and we go from orange to black. The student should use iodine drops into the water with. I think that makes sense. I should use iodine drops, yeah, with a pipette, and the water will go orange if not present to blue back if present. This is just food test stuff. I'd recommend you revise it just for the purpose of today. I'll go over them all. For starts, it turns iodine from orange to black. The sugars, we need to use Benedict solution, where it's from blue to green to yellow, but ultimately ends up with orange or brick red, and is heated in a water bath. Did I mention? Brick red, yeah, I didn't say brick red. But then protein is butyrate, biuret reagent, so BIURET, and that goes from blue to purple. Lipids, fats. You use cold ethanol, but then you mix that with distilled water and it'll turn cloudy if it is present. Those are the basic food tests. I recommend knowing them because they could come up. Basically, there's always at least one required practical coming up. And it could be this one as well. Anyway, let's go on to the next one. Buffer solution retains to its pH when small amounts of acid or an alkali are added to it. Buffer solutions can be used to keep the pH of a substance constant during an experiment. The pH of the mixture will remain constant at 5.5. So I'm reading this upside down. The student has the following buffer solution available: 5.5, 6.0, 5, 6.5, 7.0, 7.5, and 8.0. Describe how the student can adapt the Visking tube experiment to investigate the effect of pH on the action of amylase. This is very simply, very obviously an enzymes question. So all we need to do, first of all, with these described ones, you need to think about where you're getting these marks. In general, you need to state the variables. So first of all, let's state the independent variable. Now the tip is to write in full sentences. So that's how I'll be writing in full sentences. So first of all, the bullet point 1, the variables, the independent variable. So of course, if they're changing this pH, this buffer solution, then that's going to be the independent variable. It's the one you change, while the dependent variable is the one you measure. So in the independent variable in this experiment is the pH. I always do lowercase p, capital H. Well, not sort of matters though. Of the buffer solution, but that's not all. The dependent variable. So what are you measuring? You're measuring the effect, the time for amylase to break down starts. So the Venom's time taken and the control. Okay, I know it's saying it's describe how it can adapt the experiment, but generally all of this at least gets you a mark. Variables are volume of because we're adapting it to volume of water, temperature. We'll just go over those two. Okay, so now how do we adapt this experiment? So first of all, you need to look at this. Beaker water, whisking tube containing large solution amylase. To be fair, I think it's volume of I say volume of starts instead of water. Sorry, apologies. I don't really know what I'm doing. Anyway, so now we have the variables. Now I need to look at what you do with the experiment. So you have the amount of all the things. So what you would do now, since So let's measure out. So you would then place the beaker in a water bath to keep a constant temperature. I think this lip paper's taken out of me for the reaction. Okay, so you have the water bath. But whilst that's going on, you must. Oh. This isn't because the thing is, you would usually have a sporting tile, but if you want to make sure that you definitely, because this is the visking tube which contains starch and amylase. If this tube should have hopefully that, so I'm gonna change it in a way to prepare. So we're seeing how long it takes for amylase to break down. So we're looking for sugar, because once starch breaks down, it turns to sugar. So we're looking for it. So we'll be actually, well, some students may be thinking, right, we want to do starch. Starch is what's being broken down. So I guess on one hand, you could do starch, iodine, or you could do benzoic solution. Either one should be fine. Just because I guess it's amylase, I guess I'll use starch. But I don't know if you'd be discredited using benzoic solution. I'd play it safe, go for starch. But don't be confused. Even though you're using starch, we're seeing it go from or start off black, but once iodine has gone, it should turn orange. Once the starch broken out, it should turn orange because it's no longer present. So good onset. So prepare iodine drops with a pipette. Um. I think I've confused the question, but don't worry. So So the thing is with this experiment is that you need to adapt it to be different. You know, the visking tube. So what I might have to do is a bit on the fly improvement here, because I've been going on the assumption that everything in the visking tube has to remain the same, but however you can adapt it. So down here, I'll probably put you need to now you measure the the starch and amylase separately. And place amylase starch and buffer solution. I don't keep it the beaker. I guess you can if you want to keep it in the water bath. So that's what you would do there. I suppose you could also say you could put the water bath. I think that would work as well. Okay. Right. Oh yeah, hang on about it. Hang on about it. And yeah, it's you got to be ready for these questions that are going to be confusing because it's the same experiment as you would do. Just the reaction of starch to amylase is going to be within the whisking tube. This projectile, and then fill spots with the iodine. Okay, right, and then mix starch and amylase in the. Here we go. Here's where it comes together in the whisking tube, and start a start timer, and then every 10, you can say any amount of time as long as it's reasonable, 10, 20, probably no more than a minute. Every 10 seconds, remove a drop of, oh my goodness, try to be nice, kind of it, drop of the mixture and put in the spot with iodine. If the iodine changes to blue, black, the enzyme has not enzyme probably should put amylase. It's not dissolved. Actually, I'm not going to change that. Reacted with all the starts. Okay. Once all the starts. What the heck is going on there? Always some random stuff. Broken down. The iodine will remain orange. Okay. So now that's the main part. Now you have to make sure you repeat the experiment for different pHs of buffering or buffer solutions. Oh my goodness. Yeah. That's basically all you need to know. If you were asked to draw if you're asked to draw like a graph, then what I would do is down here, do there, Um, they would probably like already have the graphs labeled, but I'm guessing this would be the pHs on the x-axis. This would be the time taken on the y-axis. And what you do is you be like, okay, from here you go there, 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 there. Let's see what I've got there. I already explained this at the start, but just for those who aren't aware, and you draw a line of best fit, and the optimum lies right in between the lowest point. That would be what the graph looks like. So yeah, here we have the entire. You can note these down. So the main points is to state the independent variables. You can state the equipment. I suppose if you would state the equipment, you'd say a spotting tile, a a test tube, water bath. But I'm not sure how necessary that is. I'd just probably state it in case. So no, I just add a note. You don't have to spend too long. Oh, my setup is slowly starting to crumble. Water bath, dude. Oh my goodness. Right, this is not going to last. Good old ending stream. Dude, I really need a better way of doing this, don't I? I really need a better way of doing this. You need iodine. Of course, you need the buffer solution. Starch. And amylase. You need a pipette to measure. And a beaker. And some water. And of course, a whisking tube. There you go. Full list. So, as I said, just add that. You can just add all the equipment down there. You see all of that? Okay, nice. So yeah, here's the whole. You get state all the variables, state the equipment if you want. Then you say how you measure out the stuff with a pipette, or if it depends how big the quantities are, they'd usually say, I guess from this scenario, you'd say with a pipette, and then pipette or measuring cylinder, depending on the quantities, and then you place separately in a water bath, along with the buffer solution. You place it all separately, and then whilst that's like going for a small amount of time, you prepare a spotting tile. Then you mix the reactions together, start a timer. In this case, it was in the whisking tube, but usually it'd just be in the test tube. So in this scenario, I probably wouldn't even need the test tube. But most scenarios you would, because I guess the whisking. What is it? Oh my goodness, the whisking tube. There's like a test tube, but they act the same kind of thing. And then every 10 seconds, you move a drop, put it in the spotting tile, change the color. This starts. When it doesn't change color, no longer starts. Repeat different pHs or different temperatures, depending on what one it is. In this case, it would be pHs. And then finally, plot all the things on the graph, and the optimum condition is the two lowest points. Oh my goodness, and that is almost exactly 30 minutes. Thank you so much for being here. Good luck on your exams, guys. Please do not stress it like I am. Tomorrow morning, I'm probably going to do, a bit of RS as well. I've done some tonight. But yeah, I just looked at the cryopracticals to made a couple notes on all of them. Yeah, dude, it's nervous. It's not a nice, feeling, but this lit exam, hopefully, of course, it made me worry about the grade boundaries, but does that really matter? Everyone has done well, and to be fair, I really wish that it wasn't based on the grade boundaries, because otherwise I'd be like, over the moon for you guys. Of course, I don't like It's just the cynical nature of this whole, like, how the exams work. They kind of they base off failure, well, because you actually want other people to fail. It's kind of sad. Because of course, if I wasn't trying to get the best grades in English literature, of course, I'm doing at college, I would be like, "Come on, guys, everyone, you got this." But I still will do that, and I still make these videos because it helps me. But I hope it helps you as well. I hope that if a question comes up and it's about optimum pHs and about the whole amylase and starts and enzymes, and I hope this has done something. I hope it actually has done some stuff and food tests as well. Like, yeah, so biology will go, it'll pass. People do geography, it can't be me. They'll have their thing that happens. And then I'll just be, I'll revise maths. I think that's gonna be the scariest exam for me, maths. Top three exams. I think French listening and speaking is my most favorite one. That's the one I'll be like, yeah, even RS. I've done really well in RS. I've got mainly nines, seven, eights, nines, but that's one of my least confident ones. So I have no idea how that's happened. Maybe it's bias, maybe it's luck, maybe it's just God. Wow. Anyway, um this is the one that RS is the one exam where you can legitimately pray to the gods to help you because they probably want to. Self-centered. Sorry. Oh no, I'm gonna be cursed. Yeah, this is. Oh, I just saw this Dumbledore card. Oh, look at him. Did he disappear? I thought he disappeared. No? Okay. Okay, you don't do that then. Anyway, thank you so much for watching. My stream is about to collapse on me. Good luck, everyone. Even though it's about the green boundaries. For this instance, who cares about the green boundaries? Do your best. Do as much as you can. And I'll see you in the next stream, whenever that is.